All right, ready to dive deep into protein synthesis.
Absolutely.
I'm really excited to untangle this fundamental process, how genetic info goes from DNA blueprints to proteins in our cells. I think it'll be captivating to see how it all works.
Definitely. It all starts with the central dogma of molecular biology. So the flow of genetic information begins with DNA.
The master blueprint.
Yeah. And then it moves on to RNA.
Right.
RNA is the messenger, right? And then ultimately this culminates in the production of proteins.
The workhorses of the cell.
Okay.
So DNA holds the code, RNA carries it, proteins bring it to life. It's like a relay race, passing on genetic information.
Yeah.
But why is DNA so good at protecting this code?
Well, one key reason is DNA stability. And unlike RNA, DNA lacks a hydroxyl group
Yeah.
at the 2' position of its sugar backbone.
Okay.
And this makes DNA much more resistant to degradation.
Oh, so that missing hydroxyl group is the key to DNA's longevity.
Yeah.
But I've heard about the RNA world hypothesis, where
Yeah.
RNA, not DNA, might have been the star of the show. What's that about?
So the RNA world hypothesis proposes that early life forms relied on RNA for both information storage and catalytic activity. So imagine a world where RNA molecules, not proteins, were the primary catalysts.
Wow, so different from today.
Right.
But how did proteins take over RNA's role?
Well, proteins have incredible structural diversity, right? And that's thanks to their building blocks, the amino acids,
Oh,
and
yeah.
their unique properties.
Quite strange.
Hydrophilic and hydrophobic regions, intricate non-covalent interactions, and the ability to fold into complex secondary structures like alpha helix and beta sheet.
Those all contribute to this diversity.
Exactly, and this allows proteins to catalyze a wider range of reactions.
The proteins outmaneuvered RNA in the evolutionary race.
Yeah.
But proteins do so much more than just catalyze.
Absolutely. From enzymes to hormones, proteins are the real workhorses of life. They form structures, mediate signaling, transport molecules, and even defend against pathogens.
It's mind-boggling how much they do.
Yeah.
But let's explore how these molecules are even made. Protein synthesis is complex, right?
Oh, definitely. It's remarkably intricate, and it uses up a lot of a cell's energy.
Yeah.
In rapidly growing bacteria, almost 90% of their energy is devoted to protein synthesis.
Wow.
But it's also incredibly efficient.
I can imagine it requires a lot of cellular machinery.
Mm-hmm.
What are the key players?
Well, the ribosomes are the stars of the show, the protein synthesis factories.
Okay.
These intricate molecular machines made of RNA and protein are where the genetic code is translated into amino acid chains.
So ribosomes are where the magic happens. How do they build proteins so precisely and quickly?
Ribosomes are super efficient. They add about 20 amino acids to a growing polypeptide chain every second.
That's fast.
Imagine a bacterial cell with about 15,000 ribosomes churning out proteins at that rate.
It's like a cellular assembly line. But what about those amino acids, the protein building blocks? How do they get to the ribosomes and become part of the chain?
That's where tRNA comes in, transfer RNA.
Okay.
These small RNA molecules are adapters linking nucleic acids to proteins.
So tRNAs are the interpreters, bridging the gap between genetic code and amino acid sequence.
Yeah, exactly.
How do they do that?
Each tRNA has 2 key regions. One binds specifically to a certain amino acid and carries it to the ribosome.
Okay.
The other region, called the anticodon, recognizes a specific codon on the messenger RNA, the mRNA molecule.
So that ensures the correct amino acid is added to the growing polypeptide chain.
Right.
It's like a lock and key. But hold on, how does the cell know where to start and stop reading the genetic code? It's a lot of information.
Yeah, it's like a library.
Right.
That's where open reading frames come in, or ORFs.
Okay.
ORFs are DNA segments with instructions for building a protein. They start with AUG, the start codon,
Which codes for?
methionine, and they end with a stop codon, which signals to stop protein synthesis.
So those codons are like punctuation marks.
Yeah, they guide the machinery to the right reading frame within the genetic code.
What about errors? Mutations can mess up the code, right?
Definitely. Mutations are changes in DNA sequence that can come from errors during replication or exposure to mutagens.
And they can have all sorts of effects.
Right, from harmless changes to disruptions in protein structure and function.
Like a glitch in the matrix.
Yeah.
How can mutations change the reading frame and the protein?
One really bad type is a frame shift mutation. This happens when nucleotides are inserted or deleted from the DNA, shifting the reading frame and changing the whole amino acid sequence.
It's like a typo that ruins the sentence.
Exactly.
Are all insertions and deletions equally bad? What if 3 nucleotides are added or deleted?
Good question. If it's a multiple of 3, it won't cause a frame shift because the reading frame stays the same.
Oh, okay.
However, the protein will have extra or missing amino acids.
And the impact depends on.
The location and function of those changed areas.
Fascinating. If frameshift mutations scramble the genetic message, can it be fixed?
Yes, suppressor mutation can counteract a frame shift mutation.
Oh.
It can add back a deleted nucleotide or delete an inserted one.
So it's like a genetic do-over.
Yes, putting the reading frame back on track.
But how did scientists figure out this code in the first place? Linking codons to amino acids must have been like cracking a secret language.
It was a huge effort requiring some ingenious experiments. One pioneer was Marshall Nirenberg.
Oh yeah, I've heard of him.
He did experiments that helped decipher the genetic code.
Tell me about them. How did he crack the code?
One of his key experiments used a synthetic mRNA, poly-U, made entirely of uracil.
Okay.
He mixed it with stuff for protein synthesis, ATP, GTP, an E. coli extract, and 20 amino acids, each radioactively labeled.
He simplified the system to isolate the effect of one codon. What did he find?
He found that only phenylalanine was incorporated, meaning UU codes for phenylalanine.
That's a big discovery, the first link between a codon and an amino acid.
Right. It was a major breakthrough.
What about tRNA? How did he figure out its role?
In another experiment, he looked at ribosomes and saw that specific tRNAs, each with a specific amino acid, were bound to them. This proved that tRNA links mRNA codons and amino acids.
So his experiments revealed the dance between mRNA, tRNA, and ribosomes in protein synthesis.
Exactly. It was groundbreaking.
These discoveries must have helped create a better understanding of the genetic code.
Absolutely. Scientists then made a complete codon amino acid dictionary.
Mapping all 64 codons.
Yes, to their amino acid or stop signal.
Like a Rosetta Stone for life. But I've heard that genetic code is degenerate. Does that mean it's flawed?
No, not at all. Degeneracy or codon redundancy means multiple codons code for the same amino acid. This makes it resistant to mutations.
Okay.
If a mutation changes the third base of a codon, it often doesn't change the amino acid, leading to silent mutations.
So it's a backup system.
Yes, in case of typos in the genetic code. But not all positions within a codon are equal when it comes to mutations.
Right. Some matter more than others.
The first position is most critical, then the second, and the third is usually most tolerant because of degeneracy.
It's like a hierarchy of importance within a codon. What if a mutation does change the amino acid sequence?
Those are called missense mutations, and their effects vary. Sometimes the new amino acid is similar and it causes minimal functional changes.
What other time?
It can disrupt protein structure or activity. And then there are nonsense mutations.
Which do what?
They create a premature stop codon, leading to truncated, often non-functional proteins.
So they're like sudden roadblocks.
Yeah.
Are there ever variations in the genetic code that are good?
Yes. The mitochondrial genome has its own slightly different code. In mitochondria, UGA, normally a stop codon, codes for tryptophan.
Interesting.
And AGA and AGG, normally arginine, act as stop codons.
Huh.
This difference shows the evolutionary divergence of mitochondria.
So even within one organism, there can be variations.
Yes, it's fascinating.
But if tRNA and mRNA interact so precisely, how can those third base variations happen?
That's where the wobble effect comes in. The interaction between tRNA's anticodon and mRNA's codon isn't perfectly stringent, especially at the third position.
So there's some wiggle room.
Exactly. For example, codons, starting with CG, compare with the tRNA with the anticodon 3' GCI, where I is inosine.
Inosine? What's that?
It's a modified nucleoside that compare with A, U, or C.
So the wobble effect allows variation without messing up protein synthesis.
Precisely.
We've been talking about point mutations, single base changes. Are there other types of mutations?
Yes. Point mutations can be transitions or transversions. Transitions happen when a purine is replaced by another purine or a pyrimidine by another pyrimidine.
And transversions.
They involve a purine being replaced by a pyrimidine or vice versa.
Are there other cool things that challenge the rules of translation?
Yes. There's something called translational frame shifting.
What's that?
Ribosomes can slip by one base during translation.
Really? That seems like it would mess everything up. How does that happen? And what are the consequences?
Well, it's not random. Translational frame shifting is often triggered by signals in the mRNA sequence.
Like what?
A common one is a combination of mRNA secondary structures like pseudoknots or stem loops and slippery sequences.
What are slippery sequences?
They're usually AU-rich regions with weaker base pairing.
Making it easier for the ribosome to slip.
Exactly.
So the ribosome hits this roadblock in slippery terrain, stalls, and then shifts its reading frame.
Yes, like a molecular detour.
But why would this be helpful?
Well, consider viruses. They have compact genomes. Frame shifting lets them encode more proteins from a limited amount of genetic material.
Like packing more into a small suitcase?
Right, it expands their protein repertoire.
Are there other examples of frame shifting?
Yeah, it's been found in bacteria, yeast, even humans. It might regulate gene expression and create protein diversity.
Wow, so it's not just a viral trick.
Right. And remember those rare codons?
Yeah.
They can influence frame shifting too. When a ribosome hits a rare codon, it might stall, waiting for the matching tRNA.
And if it waits too long?
It might skip the codon, shifting the reading frame.
Like improvising.
Exactly.
Let's take a closer look at what controls translation. What else is involved besides ribosomes and tRNAs?
Initiation factors are really important.
What do they do?
They help assemble the ribosome on the mRNA and make sure translation starts at the right place.
Like stage managers?
Yes. They make sure everything's set up correctly.
What do they do specifically?
There are different ones with different jobs. For example, IF2 in bacteria helps bind the initiator tRNA, carrying methionine, to the small ribosomal subunit.
Yep.
Then IF3 prevents the small subunit from binding to the large subunit too early. And IF1 makes sure the initiator tRNA binds to the correct site on the ribosome.
They work together to start translation smoothly.
Exact.
How does the ribosome move along the mRNA once it's all set up?
That's where elongation factors come in.
What do they do?
They're essential for speed and accuracy. They help deliver tRNA molecules to the ribosome.
Okay.
They also help form peptide bonds between amino acids and move the ribosome along the mRNA.
They're like the choreographers.
Yes, guiding the tRNAs and making sure the steps are done right.
What happens in elongation?
It's a cycle with a few coordinated steps.
Yeah.
First, a tRNA with its amino acid binds to the A site of the ribosome.
Okay.
This is helped by EF2 in bacteria, which uses GTP as energy.
So the first step is matching the tRNA to its codon.
Right.
What's next?
A peptide bond forms between the amino acid on the tRNA at the A site and the growing chain on the tRNA at the P site.
Okay.
This is catalyzed by peptidyltransferase,
Which-
part of the large ribosomal subunit.
So the chain grows by adding the new amino acid.
Yes,
What happens to the tRNA at the P site?
the ribosome moves along the mRNA by one codon
Close.
translocation.
Right.
This is helped by EFG in bacteria.
All right.
During translocation, the tRNA at the P site goes to the E site and is released.
And the one at the A site.
It moves to the P site, now carrying the growing chain.
It's a cycle of binding, bond formation, and translocation.
Yes. The ribosome keeps moving, adding amino acids one by one.
This can't go on forever, right?
You're right. It stops when the ribosome reaches a stop codon.
And that's where release factors come in.
Exactly. They recognize stop codons and trigger the release of the polypeptide chain.
Like a curtain call.
Yes.
How do they stop translation?
They bind to the A site when a stop codon is reached.
Okay.
They then promote hydrolysis of the bond between the last tRNA and the polypeptide chain.
So the chain is freed?
Yes. And it can fold into its proper structure. Then the ribosome detaches and its parts can be used again.
It's amazing how regulated the whole process is.
It is.
What about errors during translation?
They can happen, leading to the wrong amino acid or premature termination.
But the cell can fix them.
Yes, it has quality control to detect and fix errors.
Like what?
Ribosomal proofreading is one.
What's that?
Ribosomes can remove incorrect tRNAs before the wrong amino acid is added.
Do they double check their work?
Yes. There are other quality control mechanisms too.
Right.
Some involve proteins that check protein folding.
To make sure they have the right shape.
Right. Others target misfolded proteins for degradation.
Wow. This must all take a lot of energy.
It does.
Where does the cell get the energy?
Mostly from ATP.
ATP powers all this.
Yes. ATP is used at many steps, like for the elongation factors EF2 and EFG, and for aminoacyl tRNA synthetases.
It's amazing how ATP fuels this whole process. Protein synthesis is really dynamic.
Yes, it is.
There's so much going on.
We're still learning a lot about it. Scientists are researching frame shifting, tRNA modifications, and quality control mechanisms.
There's still so much to discover. This deep dive has been incredible.
I agree.
It's given me a new perspective on the complexity and efficiency of protein synthesis. Zooming in on the details has made me appreciate the bigger picture.
Yes, it's like exploring a city and then stepping back to see it as a whole.
Exactly. Protein synthesis isn't just about making individual proteins, it's about orchestrating life.
Precisely.
Oh.
And it's always adapting to the cell's needs.
Through transcription and translation, DNA is transformed into all these proteins essential for life.
Right.
The diversity is just amazing.
It is. And each protein's function depends on its 3D structure, which is determined by its amino acid sequence, which is determined by the genetic code.
It all goes back to DNA. But what about when DNA is altered?
Well, mutations can have various effects.
Ranging from.
Silent changes to severe alterations. Luckily, cells have ways to deal with them.
Like what?
DNA repair pathways and quality control.
It's like they have mechanics constantly making repairs.
That's a good analogy. But sometimes errors slip through.
And those can be good.
Yes, mutations create genetic variation, which is crucial for evolution.
So even mistakes can be beneficial.
Absolutely. They're the source of diversity and adaptation.
That's incredible. But manipulating the genetic code brings up ethical questions.
It does. As we learn more, we can engineer proteins and target genes for therapy, but we need to be responsible.
It's a powerful tool.
We need to be careful and ethical.
This deep dive has been eye-opening.
It has.
It's made me appreciate the beauty and efficiency of the molecular world.
I agree. And there are more wonders to discover.
Until next time.
Welcome back. We were talking about translational frame shifting, right? Where ribosomes can slip during translation.
Yeah, I was really curious about that.
So it's not random.
Oh.
It's often triggered by signals in the mRNA.
What called as signals?
A common one is mRNA's secondary structures, like pseudoknots or stem loops, combined with slippery sequences.
Slippery sequences.
Yeah, those are AU-rich regions with weaker base pairing, so the ribosome can slip more easily.
So it's like the ribosome hits this roadblock and then skids a bit.
Yeah, kind of like a molecular detour.
But why is this useful? It seems like it would cause problems.
Well, think about viruses. They have small genomes, right? Frame shifting lets them make more proteins from a limited amount of genetic material.
Oh, that's clever. It's like packing more clothes into a small suitcase.
Exactly. It expands their protein repertoire without needing extra genes.
Does frame shifting happen in other organisms too?
Yes, it's been found in bacteria, yeast, even humans.
Wow, so it's not just a viral trick.
Right. It might help regulate gene expression and create protein diversity.
Remember those rare codons we talked about?
Yeah.
Those can affect frame shifting too, right?
Yes. When a ribosome encounters a rare codon, it might stall while waiting for the right tRNA.
And if it waits too long?
It might skip the codon, shifting the reading frame.
So it improvises a bit.
Exactly.
So besides ribosomes and tRNAs, what else controls translation?
Well, there are initiation factors.
What do those do?
They help the ribosome assemble on the mRNA and make sure translation starts at the correct start codon.
So they're like the stage managers, getting everything set up properly.
Yeah, that's a good analogy.
What specifically do they do?
There are several different ones, each with a specific job.
Okay.
For instance, IF2 in bacteria helps bind the initiator tRNA, which carry the methionine, to the small ribosomal subunit.
Okay, what about the other one?
IF3 prevents the small subunit from binding to the large subunit too early.
And what does IF1 do?
IF1 helps make sure the initiator tRNA binds to the right spot on the ribosome.
It's like a well-coordinated team.
Yeah, they make sure translation starts smoothly and accurately.
Okay, so once the ribosome is assembled and ready to go, how does it actually move along the mRNA?
But that's where elongation factors come in.
The elongation factors, okay?
They're really important for the speed and accuracy of translation.
How so?
Well, they help deliver tRNA molecules to the ribosome.
Each carrying its specific amino acid.
Exactly. And they also help form peptide bonds between the amino acids, and they help the ribosome move along the mRNA.
So they're like the choreographers, making sure everything happens in the right order and at the right time.
That's a good way to put it.
So what are the steps involved in elongation?
It's basically a cycle with a few key steps.
Yeah, okay.
First, a tRNA carrying an amino acid binds to the A site of the ribosome.
Go ahead.
This binding is helped by EF2 in bacteria, which uses GTP for energy.
So the tRNA matches up with its codon on the mRNA.
Right.
Then what?
Then a peptide bond forms between the amino acid on the tRNA at the A site and the growing polypeptide chain that's attached to the tRNA at the P site.
So the chain gets longer.
Exactly. This reaction is catalyzed by peptidyl transferase.
Peptidyl transferase.
Yeah, it's part of the large ribosomal subunit.
Okay. What about the tRNA that was at the P site?
After the peptide bond forms, the ribosome moves down the mRNA by one codon.
This is called?
Translocation.
Translocation, right?
Yeah. And this is helped by another elongation factor, EFG, in bacteria.
So during translocation.
The tRNA that was at the P site moves to the E site and is released from the ribosome.
Yeah, the tRNA that was at the A site.
It moves to the P site, and now it's carrying the growing polypeptide chain.
It's like a constant cycle of binding, bond formation, and translocation.
Yeah, the ribosome keeps moving along, adding amino acids one by one.
But eventually it has to stop, right?
You're right. The process ends when the ribosome reaches a stop codon on the mRNA.
And that's where release factors come in.
Exactly. Release factors recognize the stop codons and trigger the release of the finished polypeptide chain.
So they're kind of like signaling the end of the show.
Yeah, that's a good way to think about it. They bind to the A site when the ribosome hits a stop codon.
And then,
They help break the bond between the last tRNA and the polypeptide chain.
so the protein is finally free.
Yep, and then it can fold into its final three-dimensional structure.
And the ribosome?
It detaches from the mRNA, and all the parts can be recycled for the next round of protein synthesis.
Amazing how efficient it is.
Yeah, it's a really well-organized process.
But what if there are mistakes during translation?
Well, errors can happen.
Like what kind of errors?
Sometimes the wrong amino acid might get added to the chain.
Or...
Sometimes translation might end too early.
But the cell has ways to deal with these, right?
Yes. Luckily, there are quality control mechanisms that can detect and fix these errors.
The kind of quality control.
One important one is ribosomal proofreading.
Ribosomal proofreading.
Yeah, the ribosomes themselves can actually check for mistakes and remove incorrect tRNA molecules before the wrong amino acid is added.
So they double check their own work.
Exactly. And there are other quality control mechanisms that happen at different stages of protein synthesis.
Well, like what?
Some involve proteins that make sure newly synthesized proteins fold correctly.
To get the right shape.
Right. And others can target misfolded or damaged proteins and break them down.
So they get rid of the bad ones.
Exactly. That prevents them from building up and causing problems.
This whole process must require a lot of energy.
It does.
So where does the cell get all this energy?
Well, the main energy source is ATP.
ATP. Okay. And it's used for?
Lots of things.
Yeah.
For example, ATP is needed for the elongation factors, EF2 and EFG.
Which do what again?
They help with tRNA binding and ribosome translocation, remember?
Oh, yeah. Wait.
And ATP is also used by aminoacyl tRNA synthetases,
And those are...
the enzymes that attach the amino acids to their tRNA molecules.
It's amazing how ATP powers all of this, making sure the genetic code is translated accurately into proteins.
Yeah, it's a vital part of the process.
Protein synthesis is so dynamic.
It-
And there's still so much we're learning about it.
Absolutely. Scientists are still investigating all sorts of things like translational frame shifting, modifications to tRNA molecules, and how all the different quality control mechanisms work together.
It's an exciting field of research.
It really is. There's still so much to discover.
Yeah, it makes you realize just how much is happening inside every cell all the time.
It's really remarkable.
It's like focusing on all the details makes you appreciate the bigger picture even more.
Right, like we've been looking at all the individual parts, the factories and the workers, and now we're stepping back to see the whole city.
Exactly. Protein synthesis isn't just about individual proteins, it's about orchestrating all of life.
It is, and this whole system is constantly adjusting to the cell's needs and the organism's needs.
Right.
Through transcription and translation, the DNA code is turned into this huge variety of proteins that do basically everything for life.
It really is amazing how many different proteins there are.
It is.
And they all have their specific jobs.
And each protein's function is tied to its 3D structure, which comes from its amino acid sequence, which comes from the genetic code.
So it all goes back to DNA.
It does.
But what about when the DNA code is changed?
Yeah.
Like by mutations?
Well, mutations can have all sorts of effects, as we talked about.
Right, from silent ones to really harmful ones.
But cells have ways to deal with them.
Like what?
They have DNA repair systems and quality control mechanisms during transcription and translation.
So they're always trying to keep things running smoothly.
Exactly, like a team of mechanics constantly making repairs.
That's a good analogy.
But sometimes errors slip through.
And sometimes those errors are actually good.
Yeah, sometimes. Mutations are what create genetic variation.
Okay.
And variation is what drives natural selection.
So we need mutations for evolution to happen.
Exactly. Without mutations, there would be no adaptation, no change, no new life forms.
That's amazing. But what about when we intentionally change the genetic code? Like for research or for therapies? Doesn't that bring up some ethical issues?
Definitely. As we learn more about the genetic code and protein synthesis, we're getting better at engineering proteins and targeting genes for therapy.
Right.
But we have to be very careful about how we use this knowledge.
It's a powerful tool.
It is.
Oh,
We need to have open discussions and be transparent about what we're doing, and we always have to keep ethical principles in mind.
It's a lot of responsibility.
It is.
This deep dive has been incredible. It's made me think about things in a whole new way.
I agree.
It's amazing how much we've learned about the molecular world and how much more there is to discover.
Definitely. There's always something new to learn.
Until next time, everyone, keep exploring and stay curious.